have Tamara Stawicki.
Stawicki. Oh, that's loud. I'm from New Jersey. Sorry.
In New Jersey.
Oh, well, right now I'm in Seattle. So thank you for that introduction, and I'd also like to thank the organizers for giving me the opportunity to talk today. So I'm going to be continuing to tell you about hair cells, and Mark already did a great introduction, but just as a reminder, for those that came in late, we're not talking the hair on our head, but rather these cells that in humans are in the ear and important for auditory and vestibular function. And the problem is, like Mark mentioned, a number of things can either kill or damage these cells. And these include loud noises, the simple act of getting old, certain medications and toxins, and there's also a number of genetic disorders that lead to hair cell dysfunction. So I really came into this project interested in how can we prevent these cells from dying in the first place. And what I'm going to tell you about today specifically are these medications, and namely these aminoglycoside antibiotics. And to do this, we're going to look at the zebrafish lateral line system. So this is a 5-day-old fish that is expressing GFP in hair cells. So up top, you see the little dots along the length of the fish are these little clusters of hair cells. And down below, we have kind of a zoomed-in version. I guess we don't have a mouse. But if you look at the image on the right, you'll see the bottom is your cell bodies. That smaller, kind of dark green are the stereocilia, and the longer green is the kinocilia. And just as relevant to this talk, even though the stereocilia is called cilia, they're not actually cilia, they're rather actin-based structures. So if you look at the other image, you'll see if we stain for actin, we're labeling that lower structure, whereas the taller structure is the actual true cilia of the hair cells, which is the kinocilia. Oh, there's my mouse. Oh, there my mouse went. Okay, so if we're going to talk about aminoglycoside hair cell death, it's obviously important that the aminoglycosides killed the hair cells in the lateral line, and that does indeed appear to be the case. So on the top here, we have a wild-type fish like I showed you, and the bottom is a fish that's been treated with 200 micromolar of neomycin. So you can see in that case, the hair cells have been lost. Now, one of the advantages of using the zebrafish is that we can perform these genetic screens. So we've been screening, looking for mutants that are resistant to aminoglycoside-induced hair cell death. So I have a mutant on the bottom here that's also been treated with 200 micromolar of neomycin, but now you see the majority of the hair cells remain. So we've been doing this for a while, and there's 5 mutants where we've gone ahead and found the genes, and interestingly, they fall into 1 of 2 classes, either genes important for global pH regulation or genes important for cilia. And as you might have guessed from the title of the talk, I'm going to be telling you about these cilia genes today. We were actually kind of surprised to see these cilia genes. So a lot of people study cilia. They're involved in these human disorders called ciliopathies. But the hair cells, even though they have these big, beautiful cilia, people have mostly ignored them because auditory hair cells in mammals lose their cilia during development, so people figure cilia don't matter. The vestibular hair cells keep them, though. The zebrafish hair cells, like I showed you, still had them. So clearly, and we found them in this screen, so clearly they're doing something, so we were really interested in figuring out what they're actually doing. Now, in addition to the mutants we found on the screen, we've also looked at some existing zebrafish mutants, as well as generated some mutants, and found a number of other cilia genes that similarly appear to be important for this aminoglycoside-induced hair cell death. And I'll tell you more about these genes as we go. Okay. So the genes we found fall into 1 of 2 classes. The first class being these intraflagellar transport, or IFT genes. So cilia don't make their own proteins, they have to transport everything to the cilia, and then the IFT pathway is transporting things throughout the cilia. So you have anterograde IFT that's going from the base to the tip of the cilia, and retrograde that's going back from the tip to the base. The 2 genes we identified from the screen are both retrograde IFT genes, so there's dynein 2 heavy chain 1, which is a motor protein, and WDR35, which is part of a number of adapter proteins that link the motor to the cargo. The other 2 genes we've looked at were existing zebrafish mutants in IFT88 and TRAF3IP, and these are both mutants that are part of the anterograde version of that adapter complex. And so first, just looking at the cilia in these mutants, on the left I have a wild-type neuromass stained with acetylated tubulin. You see those nice long kinocilia coming out of the hair cells. And the other images are the various mutants, and you see the kinocilia are essentially absent in all these IFT mutants. Now, if we look at the actual kind of hair cell numbers phenotypes, so I'll walk you through this graph because there's a lot. So we're looking on the y-axis, hair cells per neuromast. The left-hand side are control hair cells, so they've never seen neomycin. The black bar is a representative wild type, and the different colors are the different mutants. So you'll see in all these mutants, we're actually seeing a slight decrease in control hair cell numbers. Now, if we look on the right-hand side, following 200 micromolar neomycin, you'll see in that wild type black bar, the majority of the hair cells have been killed. However, in the mutants, we're seeing a very significant protection. And I'd like to point out, because it becomes relevant later in the talk, the WDR35, which is that light blue, protects just as well as those other IFT mutants. So that's our IFT mutants. The other class of genes we found were transition zone mutants. So the transition zone is an area at the base of the cilia that's important for determining which proteins get in and out of the cilia. So the gene we found in the screen was CC2D2A, which is actually the first gene we identified from the screen. And since then, we've generated mutants in 2 different genes known to interact with CC2D2A, and these are CEP290 and MKS1. And it's worth noting CEP290, in addition to interacting with CC2D2A, is probably more basal body localized than transition zone. Now, if we look at the cilia in these mutants, we see something very different than what we saw in the IFT mutants. Namely, the cilia appears grossly normal in all these different mutants. And if we look at hair cell number, we're also seeing a slightly different phenotype. So again, on the left, we had the control, with the black bar being wild type and the colored being the various mutants. And you're not seeing this difference in control hair cell number in these mutants. And if we look on the right, following 200 micromolar neomycin, you're seeing that they are all protected, but it's nowhere near as dramatic a protection as what we were seeing in these IFT mutants. Now, I should note, not every single cilia gene is involved in hair cell death. So we looked at R13B, which was an existing zebrafish mutant that has some very striking cilia phenotypes and other cell types. But looking at the hair cells, if we look on the bar graph on the left at either control hair cell number or following neomycin, we're not seeing any change. And if we look with the acetylated tubulin, there's maybe less or shorter kinocilia, but the kinocilia are definitely still present. We've also looked at a number of other cilia genes, so MPHP4, RAB8, RPGRIP1, and RPGRIP1L, that similarly don't seem to show any phenotype. I should note for those other ones, we don't really see any, we haven't looked at everything, but we don't see any obvious phenotypes, so we can't say for certain that they're true mutants. It might be, we've heard plenty of talks about compensation, alternate splicing, and we don't know if any of that's going on in those guys, but they don't seem to have any hair cell phenotypes or any body morphology phenotypes. Okay. So now the big question we asked after we found all these mutants is, what are they actually doing to make these hair cells resistant? And one thing we found in a number of our mutants we've previously identified is that they affect the uptake of the aminoglycoside into the hair cell. So to look at that, we can take the neomycin conjugated to a Texas red and actually visualize it in the hair cells. So on the right hand, I guess it's your left. So on the left-hand side, we have our wild-type hair cells that have been treated with this neomycin Texas red. And you see it gets into the hair cells, it labels them quite well. On the right-hand side, we have the mutants. So up top, we have the dynein, which is one of those IFT mutants. And you'll see in that case, there is a significant reduction in the amount of the aminoglycoside getting into the hair cells, even though some is definitely still getting in. And then on the bottom, we have the CC2D2A mutant, which is one of those transition zone mutants. And here, we're not really seeing a dramatic reduction in the amount getting into the cell body, even though if you notice, it's hard to tell in the graph, but we do actually see a slight reduction in the amount getting into the kinocilia. So I have that quantified for all our mutants here. So if you look at the IFTs, so this is showing you just the data from the mutants as a percentage of the amount we see getting into the controls. So the dashed line is 100%, so if they looked identical to the controls. And you see that all of the IFTs are showing some level of reduction as compared to control. However, that reduction doesn't seem quite as strong in that WDR35 mutant, suggesting something different might be going on there as compared to the other IFT mutants. Now, in contrast to this, if we look at the transition zone mutant, we're not seeing a significant reduction in the amount of the neomycin getting into the cell bodies, at least. Okay. So just to summarize what I've told you, that we found through this genetic screen that mutations in both the Cilia IFT and transition zone genes can lead to resistance to neomycin-induced hair cell death, and that it appears that in the case of the IFT mutants, but not the transition zone mutants, we're also seeing these additional phenotypes, including a decrease in control hair cell numbers, a loss of the kinocilia, and defects in the neomycin loading into the hair cells. Now, you might have noticed I still haven't really told you how these cilia genes are protecting hair cells against neomycin, and that's really what I'm planning on doing moving forward, and just to share a couple of the ideas that we're interested in pursuing. So first, I didn't show you the data for the sake of time, but for the IFT genes, with the exception of WDR35, in addition to the neo uptake, we're also seeing defects in FM143 uptake, which is often used as a proxy for mechanotransduction activity. So we think it's possible these hair cells are not quite as active, and we're curious why that is. And one potential thought is that in addition to their role in cilia, so it's known the kinocilia itself is not important for hair cell activity, but the hair cells themselves are filled with microtubules. And there is one paper that suggests IFT88 might be working in the cell body to traffic some of these genes important for hair cell mechanotransduction activity. So we definitely want to investigate that further if some of these other genes are acting similarly. For WDR35, which is that mutant that doesn't show the hair cell activity defects and also doesn't show quite as much neomycin reduction, so we think maybe they're doing something in addition to the uptake. There's a paper from Cell Culture suggesting this gene is important for mitochondria cell death signaling pathways. So we want to see if that's happening in hair cells. And lastly, we've looked kind of at gross levels of aminoglycosides getting into the hair cells, but we want to look more closely on where they're going once they're in the hair cells and if that's different in some of our mutants. And again, trying to figure out what's going on with that difference in the kinocilia labeling in those transition zone mutants. And with that, I'd like to thank the people that worked with me. So a number of people from Dave Rabel's lab have worked on this screen and have worked on helping to map these mutants. We got a number of mutants from Cecilia Mowens, and we also got fish strains from other people as well, and an IDCD for providing my funding. Thank you. With that, I'll take any questions.
So over here.
Yeah.
So I know you're probably familiar with Maureen Barr's work showing in C. elegans at the tips of cilia, they seem to be excreting exocytotic vesicles. I'm wondering if there's something similar going on here, and if you've lost the ability to bleb off the membrane, and that, I know it's sort of the opposite direction, I can't get my head completely around this, but trying to think about why they can't take up the neomycin, and I'm wondering if we've lost some sort of endocytic, exocytic compartment at the tip of the cilium.
Yeah, so that's a good question, and we've actually never looked at that, but we have, I don't know. I'd have to think about how we could look at that. I can say in terms of the IFT mutants where we see the FM uptake, so it's known if hair cells don't mechanotransduce correctly, they're not going to take up neomycin. And since those hair cells are also not mechanotransducing correctly, we think somehow that's why they're not taking up neomycin. But whether that has to do, it would be really interesting to look at the kind of, I should think about more how to look at global excitosis. Yeah, it's not known if these cilia are blebbing things out, to the best of my knowledge.
And what do you know about what polycystins might be in these cells?
They are there, and someone knows exactly which. I know people have looked at exactly which ones, but I don't know off the top of my head, but at least one of them is definitely expressed in these cells. Yeah.
I have a quick question for you, this one. So the kinocilium is not really a primary cilium, it's really a modified motile, so it's a 9+2 very long cilium. I have been wondering, are there true primary cilia, so 9+0 little short sensory cilia on hair cells? Are we sure they're not present?
I don't think anyone's ever, well, so the lateral line hair cells, I don't think anyone's ever seen shorter cilia. And it's also, I've read a lot of EM papers on various hair cells, and they see, and I don't remember all the individual data, but they do definitely see different systems, see different microtubule arrangements in the kinocilia. So it's not that they're all 9x2. There definitely are some that don't have those internal microtubules, but I can't remember offhand which systems that was, but I do know there's a lot of variability.
Well, in fish, they're 9+2. They also have radial spoke components, so they look more motile just than both.
Okay.
All right, thanks very much.